Welcome to Ms. Chiao's video lecture in the unit of protein synthesis. We are going to be working on the third and last part, translation. Okay, let's go ahead and talk about translation as an overview. Now let's remember what happens in translation as a general overview is that you have your mRNA sequence here. As the mRNA sequence is moving, you have your E, P, and A site. So what happens is it reads the tRNA pieces. These are transfer RNA. This big orange, this big green thing here, the top part is the large subunit of the ribosome. The bottom is the small ribosome. And what happens is that a tRNA will come with the anticodon matching the codons in mRNA sequence, and it carries an amino acid. Okay. Now, another codon as it reads, now this is kind of going in the opposite direction for this picture, is that another tRNA that matches the codon here will come down. These are the amino acids. The anticodons will match the codons in mRNA. As it lands here, it is going to transfer onto the growing amino acid sequence here. So as it adds on the amino acid sequence here, it is going to be forming the new protein. And once it's transferred over, and each time it's transferred at the P site, is where the transfer RNA, now that it does not have an amino acid anymore, it will go off back into the cytoplasm. So it's almost like an assembly line. One will come, the amino acid will pop off and add to the protein sequence, the growing amino acid, the empty tRNA will fly off, and then another one will land. So as it's going along, how do we know which one? It's because the anticodons need to match with the codons. So for every 3 codons on the mRNA sequence, it equals 1 amino acid. In total in life, there are 20 amino acids, essential amino acids that our body needs. Here is a codon chart where the first base are here on the left-hand side, second base on top. So how do you read it? Well, for every U here, it's going to give you U, which codes for phenylalanine, one of the codons. So, in total, there should be 20 amino acids. We're not going to count them, but there are 20 amino acids. But you're like, there's more than one stop codon for each amino acid. So, phenylalanine has 2. There's more than 1 codon per amino acid. Why does that happen? Because each amino acid can have up to 2 to 4 codons. Let's go ahead and go back to the chart and see why. So over here, phenylalanine has 2 stop codons, or not 2 codons. Leucine, however, has 1, 2, 3, 4, 5, 6. Actually, more than 4. Leucine has the most. So it goes to say, leucine is probably a very important amino acid. If there so happens to be a mutation, let's go ahead and look at the code. If leucine has a UUA or a UUG, if there's a mutation that happens that A turns to G, it's safe. It gets to be an amino acid. Now what if it turns into UUC? It turns into phenylalanine. If it turns into phenylalanine, then depending on the protein and the properties of the folding, it may or may not be a mutation that's big enough that causes problems. So there are several things that will help give insurance to the protein. One is that for each amino acid, there's more than 1 codon for it. So if it happens to be a point mutation, then which means one type of mutation, then it's kind of like having insurance, so it's safe. So if leucine seems to have a point mutation here at the last codon here, CUU, and the U changes to C, then it's safe and becomes one. It's still leucine, one that still codes for leucine. The second thing that makes an insurance are the properties of these amino acids. If you look on top of here in the chart, the yellow means that these amino acids have polar properties. Now remember, back in chemistry, non-polar, yellow is non-polar, which means it does not like water. Polar means it likes water. And then we have acidity, whether it's basic or acidic. Now, if you look at the codes in the first column, from phenylalanine, leucine, isoleucine, methionine, and valine, these here have very similar properties. And if it had stop codon, hoping that its similar properties won't cause much mutation in the protein. Once the protein's mutated, then you'll have diseases and that may or may not be life-threatening, depending on what type of disease it is, and hinders function. But then you have polar properties, which are kind of spread out. So depending on the properties, you may or may not get a mutation that is nonsense, which nothing happens. As a note here, methionine, this is the start codon. This is, besides an amino acid, it's also a signal to start. And there are 3 different stop codons: UAA, UAG, and UGA. So each amino acid actually can have, I said 2 to 4, but technically it can be 2 to 7. On average, it's 2 to 4. But as you saw in leucine, it has 7. So in the first step of translation, now that we understand about codons, is initiation. So what happens in initiation is a small subunit of rRNA binds to the mRNA binding site. So here, the tRNA, which will carry the anticodons of UAC, will match with the codon sequence AUG. So if you were looking at the chart and you were finding out which amino acid goes here first, you have to read the codon sequence on the mRNA sequence. The codon region here will tell you which amino acid it will code for, and that amino acid will be attached to a transfer RNA, which will have the anticodon to match. Now, AUG is the start codon, and it's a place where it gets the tRNA with matching UAC sequence, as I said before. Now, in ribosome RNA, the rRNA, there are 2 parts. So the first part that binds on is small subunit. When there's the second one, the large subunit comes in. That's in step 2. The binding of UAC tRNA to AUG on mRNA signals the rRNA subunit to come. So, in step 3, the large RNA binds onto the mRNA complex. So now that the large subunit comes in, the ribosome is going to be complete. Now on this RNA sequence is going to be different sites. The E site, P site, A site binds onto the mRNA sequence. So these are additional proteins that binds on. The P site contains the area where the start codon is. So it's actually going to be here in the middle. That's where the proteins bind on here. The A site is here, and then the E site is going to be behind it. So here it is, the visual. You have the initial start codon, and that's going to be where P site, A site, and the E site. These are additional proteins that provides areas in which tRNA should be at, so there's a little bit more structure. The second part is elongation. So now that the translation has started, how do they add on more amino acids to continue forming the protein? Here in our notes, amino acids is annotated or noted as A.A. Protein starts to add more amino acids by going to the next sequence after AUG. So what's after AUG? Behind AUG is where it is. So GUC codon corresponds to valine. So this picture here, after AUG, you have GUC. So what's after GUC? And that codes for valine. So a tRNA of valine will come and land right next to the P site and the A site. So when that happens, the start codon methionine amino acid is then transferred to valine tRNA, and then it forms a peptide bond. So here is the start codon. Methionine will go ahead and transfer its amino acid. Now that it has a fellow tRNA, a tRNA right next to it, it's just going to transfer to its amino acid and form a peptide bond. The RNA now will get ready for the next codon, UUC. That's the next step. So once it's bonded, it will signal the whole entire ribosomal unit that it's going to have to shift and move. So what it's going to do is it's going to shift this whole entire RNA is going to shift going from 5' to 3', going to the right. When it goes to the right, that will go to step 6. RNA movement, the whole entire unit, that's the large ribosomal RNA and the small ribosomal unit, will shift the EPA sites by 1. The P site's tRNA moves to the E site. So what happens is this whole entire thing shifts this way, so that you'll end up with the UUC in the A site, the GUC in the P site, and then now that the tRNA is in the E binding site, they realize, okay, you don't have an amino acid anymore, we don't need you anymore, and it's released. So P site's tRNA moves to the E site, A site's tRNA with the valine and methionine will move to the P site. The A site now empty with the 4th codon, UUC. And then the tRNA finale will come towards A site because now the A site's available. It is signaling for a tRNA that has the anticodon of UUC, which is AAG, and that tRNA will carry the next amino acid in, which is phenylalanine. Okay, next is termination, step 7. A site continues to receive tRNA to match a new codon. Then the P site tRNA continues to move its growing chain of amino acid to bond with new tRNA. So let's go ahead and get our picture going here. So here, as it keeps going, every new amino acid will come in at the A site. Then it's going to bond. It's going to move its amino acid over, and then this is released. E site continues to release the empty tRNA. So as it keeps on going, E site's always going to be releasing the empty tRNAs that do not have their amino acids anymore. And then the P site's going to continue adding on their peptide bonds to the A site. And then it's going to move down one and then have availability and repeat the whole process. Process repeats until it reaches the stop codon UGA. And then, so what is the last step? Release factor binds on to the stop codon. So the last one, UGA, and there's actually 3 different stop codons. In this particular example, we have UGA. So what happens at UGA, this is a stop codon. This release factor here is a signal. Now, if you notice, it does not have an amino acid. That's the end. That's it. It says, okay, there's no more. So what happens is that it will signal this to cut off. The amino and the protein will be done synthesizing. And this whole entire subunit, the large ribosomal RNA and the small subunit with the E site, P site, and A site will just release. So here in the last step, the new polypeptide chain breaks off, the RNA unit will break apart the rest of the unit, and then the protein will go into folding. There are 4 steps to folding a protein, and protein folding will give proteins its unique 3D structure, and that 3D structure will be used for communication and recognition. And that comes to our second part of this lecture about protein folding. So what happens is, once it falls apart, you have this sequence of protein. Protein just doesn't float around. It actually has to have some structure, and it'll fold into unique forms, and that unique form will be that recognition, like the key and lock of communicating with other proteins and chemicals. So the first, there are 4 structures to a protein. The primary structure are that amino acids are connected, forming a polypeptide sequence. So just like beads on a necklace, amino acids are connected to each other with a peptide sequence. Each amino acid is represented by the bead, and they will form an entire protein. The secondary structure is that when polypeptides are linked by 2 kinds of forms, alpha helix or beta sheets. So what are beta sheets? Beta sheets are basically how you have fold paper and you look at its side cross-section and it looks like a fan, up and down, just like this fan here. An alpha helix, if you're familiar with structured DNA, it is a spiral form, just like this DNA structure mimicked here in this picture. The third structure, so first you have polypeptides to connect to each other. Then they're going to either fold like this in a twisted spiral, or they're going to fold up and down, and they're going to be connected by, they're going to fold. And then the third one is tertiary structures. Now that some parts of it's not saying the entire protein will have either the alpha helix or beta structures, it's just some of them, part of a protein can have an alpha helix. Some of it's going to be beta, or maybe a whole bunch of different betas. Whatever the protein folds in that form, they're going to go to the next step, the third step, which is tertiary structure. Now that you have some that are folded, then it's going to come all together, forming a 3D structure bigger. And how they're going to form a connector is through hydrogen and disulfide bonds, and they're attracted by the secondary structure. So over here, in this lower picture, you have an alpha helix and beta sheets. In this protein here, this whole entire thing that's green, blue, and pink is entire protein. It's just a tertiary structure where you can see those spirals here of alpha helix, and then the pink is its beta sheets. Even though it's in its beta sheet, this is a tertiary structure where they can be connected together. Now the last structure is called quaternary structure. A protein has more than one amino acid chain. So a protein can be composed of various amino acids. So it can get complicated. So if you look over here in this picture, quaternary structure, you have your original tertiary structure. But in addition to that, there's another sequence amino acid chain here binding it together. So it's like putting together a 3D puzzle. Proteins can be very complicated, and any mishap of codon sequences in which if they had a mutation here, then it may or may not be able to bond with the other protein, which can cause diseases or mishaps in functions. So we won't need to know the details of how it works, but to understand that there are 4 types of primary, secondary, tertiary, and quaternary structures in protein formation. Okay. And that is it for the protein sequencing lecture. And thank you for listening.